Hi everyone, we're just going to get started in a couple of minutes. We're going to give a few more people a chance to log in. Just give it one more minute and then we'll get started. Excellent. Okay. Good afternoon, everyone. Thank you so much for joining us and welcome to our SIFT MS webinar and on the importance of measuring volatile organic compounds in agricultural settings. So my name's Cassie McCready. I'm the Business Development Manager for Livestock and Aquaculture, and I will be your host for the webinar this afternoon in partnership with Scotland's Rural College, SRUC, and Anatune. So before we begin and get underway with introducing the speakers, I'm just going to go through a little bit of housekeeping. So the session is being recorded and all attendees are on mute and in listen-only mode. Questions will be taken at the end of the presentations and you can submit these via the questions bar on the GoToWebinar panel on the right of your screen. So we would like to hear from you throughout the presentation, so please also do interact with us via the chat function. Today, we're going to be welcoming Mark Perkins from Anatune and Professor Richard Dewhurst from SRUC. So without further ado, I'd like to take the first few minutes of your time to introduce Agri-EPI to you all before we hear from our first speaker. So as I've just gone through, my name's Cassie McCready and I'm the Business Development Manager for Livestock and Aquaculture. So who are Agri-EPI? So Agri-EPI are one of 4 centres of agricultural innovation in the UK. So we're supported via Innovate UK by the Department of Business, Energy and Industrial Strategy, or BEIS, and the 4 agritech centres exist to deliver benefits to UK farming. So we work alongside 3 sister centres, the Big Data Centre of Excellence, Agrimetrics, the Crop Health and Protection Centre, CHAP, specifically focusing on crops and plants, and the Centre for Innovation Excellence in Livestock, CL, specifically focusing on livestock. So we are a membership organisation and everything we do is centred around our membership offering. So we bring together a range of stakeholders to design and develop research innovations to support sustainable growth in the UK farming sectors. So our members come from all kinds of different types of organisations and backgrounds, everything from academic institutions to farmers themselves, process and retailers and others in the supply chain, and then critically SMEs, technology developers, entrepreneurs and solution providers. So our members get access to a range of assets and services that Agri-EPI own uniquely. So we provide access to leading academic institutions, broker collaborations and build consortia for projects. We also have facilities that allow you to test and demonstrate new technologies on our farms. Excuse me. We also support members access R&D funding, so we have a team that can help you identify the right funding calls for your project and help you bid right for those proposals as well. We have business incubation facilities in the Midlands where you can register your company. We also host KE and dissemination, events and PR, and we have a team for influencing policy. So what does our network look like? If you take a look at the map on the left-hand side of the screen, you can see that we're based up and down the width and breadth of the UK. So the 4 red dots in Edinburgh, the Midlands, Cranfield and Somerset represent our hubs, and at each of these we have a major asset base, including our Northern Food Lab, our Harper Adams Dairy Centre, our Plant and Crop Phenotyping Centre at Cranfield, and our Southwest Dairy Development Centre in Somerset. The white dots represent our network of satellite farms. So this is a very unique offering, a network of 24 commercial farms that all exist ready to test, demonstrate and pilot new technologies, giving that commercial evidence and commercial relevance. So that's just a little bit of an introduction there to Agri-EPI. Please do tweet and post on LinkedIn if you're wanting to about this event, and do use the hashtag agritechenabler for all your social media posts around this event and others. Retweet and share any of our campaigns, and let us know if you'd like to support in any other way. So at this point, I'm going to hand over to Mark Perkins from Anatune. So to introduce Mark a little bit, Mark studied for a BSc degree in chemistry at the University of Southampton and followed with a PhD in electrochemistry at the same institution. So Mark has over 12 years experience working in the materials characterization unit of the Tun Abdul Razak Research Centre, or TARC, in Hartford. So prior to this, he worked as a postdoctoral research assistant at the University of Southampton, where work focused on a novel liquid crystal templating method to produce a range of nanostructured materials for a wide range of applications, including battery electrode materials, for which a patent application was submitted. So Mark joined TARC as a senior analyst running the gas and liquid chromatography section. He was appointed deputy unit head in 2004 and unit head in 2013, managing a team of 14 analysts. So Mark, whenever you're ready, please do get started.
Okay, thank you for that introduction. Yeah. So my role at Anatune is essentially as applications chemist for Sift MS, which is one of the instruments that we distribute. We're the UK and Ireland distributor for Sift MS instruments, the instruments made by Sift Technologies in New Zealand. And so a lot of my work is looking at how to utilize the instrument in new ways, interesting ways to automate applications, etc. So I've got a very broad sort of feel for what we can do with Sift MS. So nothing really specific, just how we can utilize Sift MS as a technique to help with the analysis of all sorts of diverse areas. So the first question is Sift MS. What is Sift MS? Selected ion flow tube mass spectrometry. Well, in a word, it's a real-time VOC analyzer. So it enables you to analyze volatile organic compounds essentially in real time, with no need to calibrate the instrument, no need to do any sort of background calibrations on it. It will monitor things by the second, any VOCs you're interested in looking at. So we can get some very interesting and useful data about releases of VOCs into the environment, from products, from animals, from anywhere really where VOCs are being released and are important to look at. The other point to make about Sift MS, because it's a real-time analyzer, the results that we get, we get them in a few seconds, so we don't have to wait very long, unlike chromatography, to do lots of analyses. So we get the ability to build quite large data sets. So what I'll do briefly is to go through how Sift MS works as a technique. So first of all, so overview of the technique is, so basically it's built around soft chemical ionization from a range of reagent ions which are generated from a microwave plasma. So essentially we have moist air that is then pushed through a microwave plasma, and this produces reagent ions that we can then use and select and push into a flow tube to produce our ionization. So the first thing to say is we have this microwave plasma, and it's simply moist air that is run into the system, and we get to produce 3 positive and 5 negative reagent ions. We won't talk about the negative reagent ions today, but we get H3O+, NO+, and O2+. And what we do with these is that we pass them into a quadrupole mass filter, which selects one of these reagent ions. So it's either H3O+, NO+, or O2+. And then these get pushed through the quadrupole mass filter. So we get a single beam of reagent ions, and these are then used to ionize your analytes as they come into the flow tube. So a flow tube is a real scientific term. It sounds like it's made up, but it's actually essentially a defined thing. So what we have in a flow tube is essentially a tube that's around about 50 centimeters long, maybe shorter than that, and a diameter of about 12 centimeters. And what we have is we have essentially our reagent ions come flowing into the front of the flow tube, and we have a carrier gas, normally helium or nitrogen, that flows in first into the flow tube. And this has the effect of cooling the reagent ions down to essentially thermodynamic energy. So we have very controlled soft chemical ionization. And just downstream from the carrier gas, our sample comes in from an inlet, essentially a controlled flow inlet from external, from the ambient environment into the flow tube. So we have within that flow tube our carrier gas, our reagent ions, and our sample in the flow tube. And we have a very simple reaction takes place where our reagent ions react with our analyte to form a product ion or some product ions at some reaction rate K. And all this takes place in the flow tube in about 4 milliseconds. And all of the stuff that's left behind comes out the back end of the flow tube and is then detected by a second quadrupole mass filter. So the second quadrupole mass filter counts any of the reactive products we have, counts any of the remaining reagent ions, and essentially the software in the instrument does a calculation that can turn those results, those counts on the quadrupole mass filter, into an analyte concentration. And because the chemical ionization is soft and it's also very controlled, it means we can build up libraries. And as long as we know our K value, our reaction rate value in the system, it means that we can calculate in real time the concentration of any analyte coming into the flow tube that will react with our reagent ions. And so it is a simple way of getting analyte concentration, simply using 2 quadrupole mass filters and some soft chemical ionization. And this is what makes the instrument such a fast instrument to work with, because the amount of time it takes for these reactions to take place is really short. So from that, we can basically get a number of key benefits from this. First of all, speed of the analysis. It's also highly selective because we control our chemical ionization. We know what products are going to be formed, so we can actually separate complex mixtures. It is also inherently quantitative over quite a large range of concentration. So from low ppb to parts per trillion up to several tens of parts per million in the gas phase. And it enables us to do direct analysis. So without having to do any sample prep, without having to collect samples, we can directly analyze samples in real time. So here's an example of real-time analysis. So this is essentially looking at VOC release from coffee beans being cracked. So these are individual coffee beans that are being heated, and as they get hot, they crack and release VOCs as they heat up. And you can see here that the instrument is analyzing a range of volatile compounds that are associated with the cracking and the smell of coffee beans in real time. So you can see the time axis along there up to around about 10 minutes. And so we can monitor, as I say, in real time, a whole range of separate VOCs being released as things happen. So we can monitor dynamic processes. Now, because we have 8 reagent ions and we understand the chemistry, we actually can analyze almost all VOCs you would probably be interested in. The only ones that we can't do, of course, are the major components of air, otherwise the technique wouldn't work, and carbon monoxide, but we can do all the hydrocarbons, all the oxygenates, nitrogen compounds, difficult sulfur compounds, halogens, a number of inorganic compounds such as ammonia, hydrogen sulfide, phosphine, carbon dioxide, sulfur dioxide. So we can get a full range of these compounds. And we don't have to worry about doing one set or the other. We can actually get all of them at the same time in the analysis and separate the concentrations out. And we do this by a series of chemical reactions that take place between our reagent ion and our analytes. And there's a whole series of mechanisms available to us. And these are just sort of shown on this plot here, on this table here. I mean, we don't have to go into the detail of this now, but this is where our selectivity comes from, because we are able to react different analytes in different ways and thus pull them apart using our 2 mass spec detectors. So as an example of how this happens, so let's take propanol and acetone, as we can see here up on the right-hand side. Is acetone and propanol, they're isomeric. So from a mass spec perspective, they will have the same mass when detected. But the reaction chemistries available to us with NO+ are entirely different. With propanol, we end up with a proton extraction mechanism, where our hydride extraction mechanism, where our molecular weight on our finished product is 1 mass unit smaller than the parent ion weight. Whereas for acetone, we have an association reaction where NO+ actually associates with acetone to add 30 mass units. And so by selecting the 2 masses on our second quadrupole here, we can separate what is essentially an isomeric mixture of compounds. And we can do this often for almost all mixtures we put together. We can find reaction mechanisms and product ion reaction, reagent ion analyte pairs that will give us separations that we can make on our second quadrupole. So we can make complex mixtures and separate them out in real time. The next thing to say is it's highly sensitive. So on this plot here, we're just showing butadiene being run into the instrument around about 30 parts per trillion, whilst benzene is also in the mixture at 15 parts per billion by volume. So highly sensitive in the first instance. And at around about 60 seconds, we spike up the concentration of benzene about 1,000-fold to take it up to around about 30 parts per million. So we're now approximately 6 orders of magnitude between the butadiene concentration and the benzene concentration. But as you can see, that concentration change in the benzene actually doesn't affect the underlying measurement of butadiene at all. So not only do we have a high sensitivity, we have a wide dynamic range in that we can see low concentration species in the presence of high concentration species, and those concentration changes are not going to affect the underlying concentrations of other compounds. So again, you can see that we don't have to worry about concentrating samples up or diluting samples out to get them inside the right concentration range. We can do widely separated quantitation on SiftMS. The other thing to say is, again, linear range. So this is an example of measuring formaldehyde directly in this instance, so no need to do any sort of derivatization. So this is formaldehyde, direct formaldehyde concentration across from about 10 parts per billion by volume up to nearly 20 parts per million by volume. So we're crossing about 3.5 orders of magnitude, and you can see the system is highly linear. And this is essentially measured in real time with no calibration curve. So this is the instrument doing its inherent quantitation and generating the concentrations in real time. So you can see that what we put into the instrument, we get out as a concentration measurement. It also means we can do direct analysis without sample prep. So this is an example of measuring VOCs coming off some leather samples that were used in a vehicle. So we're looking at essentially putting some leather into a vial and heating it up and seeing what was coming off. And you can see that we get quite a wide range of species that are measured exactly at the same time. So not only do we see the hydrocarbon and the aromatic compounds coming off in the left-hand side of the plot, we also see aldehydes directly analysed without the need for derivatization. We get to see some ketone materials in the middle. Acidic compounds, so the volatile fatty acids, are also detectable at the same time in real time. Some of the more difficult, less volatile compounds, as well as odorous amine and sulfur compounds, all directly measured at the same time. No need to run separate methods for it. We can actually do the analysis in the same time. And the system is equally sensitive to all of these compounds. So as you can see, a broad range direct analysis technique that makes analysis of these compounds actually relatively straightforward and simple. So the instrument comes with some very intuitive software. It enables you to do the data processing. We can view the instrument live via remote connection, so we can have this running in a remote location and follow the data coming off live. We can generate reports, we can do method development. It comes with a comprehensive compound library, so we don't have to worry about understanding the underlying reaction chemistries. It's already in the library available to you, so you can just build methods in a few minutes and analyze whatever compounds you're interested in with very little need to understand the underlying chemistry that takes place. And we also have the ability to do automated analysis, so we can actually put automated samples on top of this instrument and do what would be difficult routine analyses in the lab very much more quickly with SiftMS. So simple headspace analysis and other types of analysis you might do in a normal laboratory space. So that's just a very brief overview of the SiftMS technique. So it really doesn't go into much detail about how it works, but if you were interested in understanding how SiftMS works, there are some very good introduction webinars on Sift Technologies website. And so if you go to that web address there are about an hour long each one of these webinars, and it also covers a whole range of other application areas as well. So I would recommend actually looking at this and getting to understand how SiftMS works, because it will help with you understanding the underlying technology. So one thing to say about SiftMS is when we compare it to traditional chromatographic analysis, what is the difference between the two techniques? What makes SiftMS different to doing chromatography for your sort of analytical techniques? Well, the difference really is that when SiftMS is having a sample introduced to it, it's actually doing the analysis at the same time. Whereas for chromatography, you introduce the sample into the instrument, and then you have to wait for quite a period of time for that sample to run through the chromatography system, to run through the column, to get your analytes out. And so your time domain for chromatography techniques is actually much longer than for SiftMS. So as long as you've got sample going into the instrument will actually analyze that sample for you, which means we can actually really shrink down the time domain on our analyses. And that can have some very interesting effects on the way you do your analysis. So here, for example, it's just a sort of a mocked up scheme of doing a SiftMS analysis versus traditional lab technology. So you can see here the black line is somebody making an analysis of some compound, and Sift can do multiple analyses in the same time it takes to run a single chromatography analysis, as detailed by the red plot here. So what it gives us is the ability to see much more rich data that sits behind what would be single point analyses for chromatography. So things that are changing on time frames of a few seconds to a few minutes will get missed if we do traditional trapping and sampling by chromatography. With SiftMS, it fills the gaps in these traditional analyses, enables us to see changes in real time in a time domain that SiftMS, that GCMS or LCMS won't get you. And that does have interesting consequences for the way you view your data. I mean, a case in point being is let's say this is a plot of a analytical space where you're making measurements and you're looking at different concentrations. So you've gridded out your analytical space. It could be a field, for example, and you're making measurements at certain grid points on that field. With chromatography, because you take so long to do the analysis, your grid is actually quite crude. It's actually quite wide, and so you don't really see the detail that sits under the plot. But if you can make more measurements more quickly, you could actually reduce the grid size of your analysis and start to see detail that you haven't seen before. So as we go in and make that grid size smaller and smaller and smaller, because we're able to make more and more analyses more quickly, you start to see underlying detail in the analysis. You start to see that what looked like just a plot of red and grey has actually got some detail in it. It actually is giving you information you've missed from that wider grid size. And in fact, it gives you so much information, you can actually see a change in the middle that you would otherwise miss versus all the other plots. I'm hoping you can see the 20-mile-an-hour symbol in the middle of the 30s. So you can see that if you can make more measurements more quickly, we actually can, by shrinking our time domain, we shrink our spatial domain as well. And we get much more information, much richer data sets, much greater data density on your analysis, enabling you to see things you probably haven't seen before in the analysis that you're doing. So, you know, the fast measurements mean you can see missing detail in your analysis. So what I'm going to cover now in the second half of this really is just some very broad applications that we can do with SiftMS. In fact, this is a very small subset of really the applications that you can do is only limited by your imagination. So the first one is like fence line monitoring using SiftMS. Now this first one is actually going to be based around a petroleum plant, but in principle the same applies. If you've got, you know, you're using anaerobic digesters or you're you know, producing biogas from somewhere. I mean, we know that agriculture always gets a bad press because of the smells that are generated, and the Environment Agency actually have some issues relating to this, and they would like to be able to monitor some of this stuff that's happening. And so having an instrument like SiftMS there doing fence line monitoring, able to monitor over time changes in VOCs, whatever those VOCs are, is actually a really useful technique to have. So this is just an example of a petroleum plant in New Zealand, but as I say, VOCs are VOCs, so for petroleum reed sulfur compounds really. And you can see here that this was monitoring some VOCs over time. So the time domain along here is about a week, and you can see that periodically we're having flares of VOCs coming out, and this was causing problems with the surrounding environment because we were getting VOC releases. I mean, not very high, only ppb you can see on the axis, but nonetheless these were going above the specified limits for this plant at these times. And when it was looked at against the times that these were happening, it was discovered that there was essentially a flare-off that was happening at the petroleum plant. That there was basically a pressure build-up, and it was just essentially flaring off the excess pressure. And this was leading to these excursions away from the underlying technique that was the underlying VOC allowance that was allowed for that production facility. But you can see if we can make these measurements continuously over a long period of time, we get to understand some information about our processes that point monitoring probably wouldn't have given you, and wouldn't allow you to make these things. There may be some, you know, day-night differences that are occurring with your process that would be good to follow. Again, we can monitor much further downstream. So this is again a fence line industrial emissions application, looking for again industrial compounds, but nonetheless showing that we can see concentration changes over time and over, you know, time frames of days and days and days that we can link up with processes that are occurring at that factory site. And because we have multiple reagent ions, we can look for the same compound using different reaction mechanisms. So what may look like noise actually can be shown to be real data. This is only a single figure ppb, so it's quite possible that this was noise in the signal. But by measuring the concentrations in two different ways, we can actually see that the signal was real, and these small variations were real in that process that was happening. So again, moving on from fence line to odor monitoring as suggested, we can do, you know, continuous odor monitoring, and compounds that produce odors are traditionally difficult to analyze for. So sulfur compounds, nitrogen containing compounds, the volatile fatty acids, they're difficult compounds to analyze using traditional techniques. Sift MS gets them with no need for any sort of sample work at all. It just measures them in real time. So here's an example of a gelatin factory where there was various parts of the factory were producing gelatin, and each part was producing different odors, obviously based on the processes taking place. And by monitoring various sites and looking for the odors that were coming off, so here we've got hydrogen sulfide, trimethylamine, dimethylamine, ammonia, you could see where the odors that were producing problems were being generated, and then you could sort of basically hone in on those sources and deal with the odors at that point, and put in place mitigation to stop that occurring, and that's making it nicer for the surrounding environment. Another example is intensive chicken farming. This was done in Australia and Queensland, and in fact on the top picture here you can see they have a Sift MS in the back of a van, so they can take it out remotely and just then monitor VOCs over a period of time to understand, again, are there day-night relationships related to the odor release or some other process that they can actually mitigate for. And here is monitoring of a number of compounds, hydrogen sulfide, some mercaptan, some disulfide, trisulfide materials, the methylamines, trimethylamine. So again, difficult odorous compounds, all being measured parts per billion, and you can see changes over time. And this was during harvesting and clear out. So obviously the cleaning out of the sheds was releasing quite a lot of odor, as you would expect, but it gives you information about the processes you're carrying out. And we can separate out the compounds. So here are the amines and ammonia. You can see how it changes over time. We can separate the sulfurs from this. So again, how did the sulfur compounds spike up in this analysis? Ketones, so we'd separate the ketones out. Now, these aren't analyses being done separately. It's all being done at the same time, but we can separate the compound classes out in the data set and understand the changes that are occurring, and see if we can again relate them to processes that are occurring in that shed. And some of the aldehydes, alcohols, and volatile fatty acids, you can see, you know, the ethanol climbing up towards the back end of that process and spiking out the acetaldehyde. So again, rich data that you wouldn't get with GC or LC analysis. Will be taking point measurements at maybe 1 hour intervals. So all this sort of rich data sitting underneath that you would miss if you didn't have the ability to do real-time analysis. Again, freshness screening using Sift MS. So do things go off over time? Of course they do. Can you do things to your product to mitigate that you know, degradation process? You can if you understand what's happening. So this is a case of screening just some fish oils for oxidation processes, and propanol was the key marker. And you know, these oils were just monitored over 5 days for the production of propanol, and you can easily see that very early on in the process we were seeing propanol coming through. And again, understanding the production of this enables you to work out processes to minimize this and minimize the spoilage of your products or in your processes. Another one, we could look at a range of other compounds in that, so not just the propanol, but other aldehydes and ethanol and acetone. And you can see over time compounds you'd expect to see rising as things go off. You can see rising ethanol being formed, for example, acetone being formed. Again, it enables you to follow the processes of your products. Another example is looking at beef samples. Again, beef going off or different sources of beef. Could we take the data and separate it out in classes to understand, just by analysing small parts, we could take product A from product B, look for counterfeiting, look for freshness, look for products that are going off. And again, we can look for compounds that you would expect to see in meat spoilage. So again, things like the methanol, ethanol, acetone, but also the odorous or compounds such as the methylamines or the mercaptans or the ammonia or the indole compounds. And you can see, particularly for the rotten sample up here, about 6 or 7 from the back, that, you know, obviously, as meat goes off, it produces significant VOCs that point to spoilage. But again, this gives you an ability to actually separate products out, to look for poor quality products or to look for counterfeit products in your factories, etc. And by taking that data and actually casting it out using class projections and some PCA type analysis, it's possible to pull out just from that data the set of compounds that have gone off versus the set of compounds from one manufacturer or one supplier from a set of compounds from another supplier. So it's possible to pull apart these products, and this is all done very, very quickly. These measurements are taking place in the order of a few seconds per sample, and yet we can still pull this data apart and get some really useful information on it. So last thing before I hand over, changes in volatile during processing. So as you process products, obviously their volatile profile changes over time because you're doing things to it. And so can SiftMS be used to do this? And this data was generated by Professor Cheryl Barringer at Ohio State University. She's been working with SiftMS for many years looking at food and flavour work, so particularly flavour released in mouth and flavour released and fragrance released during processing. And here's an example of the difference between tomato juice that's being produced with peel on the tomatoes or peel removed from the tomatoes. And these compounds identified here, the hexenal and the hexanone, for example, are known to produce off flavors in tomato juice. And you can see that if you actually remove the peel first, you get a different result from removing it after you produce the juice. And this, again, leads to quality issues and quality decisions in producing products. And again, when this was viewed against odor panels and that were testing the tomato juice, we could clearly see a correlation between the production of the odor compounds that were giving off odors versus the like or dislike in the product. And another example of this being done again is looking at off flavors in mangoes. Again, looking at a different set of compounds now, so the sort of flavor compounds you expect in mangoes, such as limonene, etc., or carvol and carvone. And again, this is processing these mangoes with peel on, peel off. How do you process the products, and does it have an effect on the finished product? Again, having information about this and understanding this will lead to good quality products and actually lead you to spoiling less and having fewer recalls on your products. And so again, we can manage this against the sensory panel, and again, we get good correlation between the sensory panel. And so with that, that's a really brief overview of things you can do. As I say, it's limited by your imagination. I'd just like to acknowledge Cheryl Barringer for that last couple of slides related to the flavour, and Vaughan Langford at Sift, who did a lot of the early work I presented. And I'm sure at the very end of this, there'll be some questions. So with that, I'd like to hand over now to Richard Dewhurst for his part of the talk. Thank you.
Mark, thank you so much. That was brilliant. Thank you for the interesting presentation. I hope by this point everybody's well versed in SiftMS before we dive into a couple of sort of deep dive applications in the agriculture space from Richard. So Richard Dewhurst is Professor of Ruminant Nutrition and Production Systems and the head of the Dairy Research and Innovation Centre at SRUC. Richard joined SRUC in 2013. Having previously headed leading ruminant research units in Wales, New Zealand and Ireland. He's the author of over 120 primary research papers, received the Sir John Hammond Memorial Award in 2008 and is past president of the New Zealand Society of Animal Production and British Society of Animal Science. Richard's research has made significant contributions at the interfaces between nutrition, product composition and rumen function, notably modelling of forage composition, dry cow feeding strategies, forages and fatty acids and facility, and rumen diagnostics. So Richard, we can see your screen and it's over to you.
Great, thank you very much, Cassie, and I hope you can hear me as well.
We can hear you perfectly, brilliant.
Great, thank you very much. Good afternoon, everybody. As Cassie said, I worked for a few years in New Zealand and so had the opportunity to interact with Sift in Christchurch around about 15 years ago. So what I'm intending to do is to tell you about a range of gas analysis applications using the SiftMS linked to animal nutrition, cattle nutrition and health, and use that to illustrate the range of volatile organic compounds that can be measured at ppb and low parts per million levels using SiftMS. I think Mark has given a really great taster of the range of it, but there are some interesting applications in animal science and agriculture, and I'm sure if you think about it, there are many, many more. I just re-emphasize the fact that the analysis is rapid and easy. One of the papers was the fastest paper I've ever published. It's scary how quickly you get data in large amounts of data from the Sift MS. I'm also going to illustrate the use of biomarkers to identify targets for low-cost sensor outputs. This slide kind of illustrates my career. I've been very interested in nutrition biomarkers, trying to use clever chemical analyses of samples that we find on the outside of an animal to understand what's going on inside the animal. And over the years, we've looked at things in feces and urine and milk and blood and hair, and the breath analysis was a logical extension on that. If we think about how we get access to breath samples, then we have expensive ways of collecting and analysing breath. These are the respiration chambers at the Green Cow facility in Edinburgh, housing animals and measuring methane emissions. But there are an increasing number of locations where we're able to collect and analyse breath samples from cows in feed stations, in robotic milking stations, forage feeding stations, and even a picture here of a cow using the SF6 technique, so being able to collect breath samples in canisters on the animal's back. So both in a research setting and also in a commercial farming setting, there are locations where we're able to capture breath samples. When I was working in New Zealand, I was telling Mark earlier on, one of the things that we were doing was collecting breath from cows at the farm at Lincoln, and driving into Christchurch, taking breath samples in containers, in Tedlow bags into Sift, and putting them through their Sift MS instrument. This is a list of some of the main compounds that are present in cow's breath, just what you always wanted to know, what cow's breath was made up of. And it's interesting when you start to look at this. Some of the compounds are related to plant metabolism. I'll talk a little bit more about the green odor reactions. Mark mentioned that in relation to some of the food quality and food preparation issues, but those enzymes in grasses and plants that are producing compounds that are present in cow's breath. There are other compounds that are coming from the rumen fermentation, so microbially derived compounds in cow's breath, and there are yet more compounds that are coming from animal metabolism. So we have a representation of plant, microbial, and animal metabolism within cow's breath. So acetone and butanone coming from breakdown of body fat in the animals, amongst other places. So that piqued our interest in what was going on in the rumen, and so we did a series of studies where we looked at the components of rumen gases. So the cow's breath is partly comprised of erupted or belched gases coming from the rumen headspace, and lots of interesting potential diagnostic components in the cow's rumen gases. So you'd expect volatile fatty acids, acetic, propionic, butyric, valeric, caproic acids. We can find alcohols in the headspace gases of cow's rumens. There's acetone, there's ammonia, acetaldehyde. One of the ones that we begin to focus more on were the sulfur-containing compounds that are present in rumen gas. Hydrogen sulfide is present at pretty high concentrations in the rumen headspace gas of cows. In this study, I should have said at the outset, we were looking at samples of rumen gases collected at intervals after feeding. The animals were fed at around 8:30 in the morning, and soon after feeding you get an increase in hydrogen sulfide. and then it tails off over time, reflecting the rumen fermentation. 200 parts per million hydrogen sulfide actually is relatively modest for a cow. We had cows with more than 2,000 parts per million. You certainly couldn't live in the rumen headspace gas of a dairy cow. Other compounds, methyl sulfide and dimethyl sulfide. Now, one of the points I was wanting to make from my perspective is we were using Sift MS to identify candidate biomarkers that we would be able to use lower cost sensors. We weren't going to strap a very expensive instrument onto the back of our cows, but these are instruments that are much cheaper, designed for health and safety monitoring. So a sewerage worker, for example, would strap one of these sensors on to monitor hydrogen sulfide in their environment. And we actually, when we identified the sulfides, we started to do more work using these hydrogen sulfide meters, and explored where the sulfur was coming from in the cow's rumen, and it's ultimately coming from sulfur, particularly sulfur within the amino acids, so breakdown of sulfur amino acids, cysteine and methionine in the rumen, liberating hydrogen sulfide. So here we were fasting the cows and then giving them a source of sulfur, and we got the biggest increase in hydrogen sulfide concentration when we put cysteine into the feed, and we got a feeding different levels of cysteine. As you increase the level of cysteine, you got a higher increase in the hydrogen sulfide concentration. So this is one where we're getting to more than 1,000 parts per million in the rumen gases. Now we did some work developing a protocol to use this to look at the breakdown of protein in different dietary ingredients, and that's in the next slide. So here what we were doing was fasting the animal and then giving it a meal of a different protein source, so maize gluten feed or sunflower meal or soybean meal, and where you might typically, to look at the breakdown of protein in the rumen, look for the appearance of ammonia in the liquid phase of the rumen. Here we were monitoring hydrogen sulfide in the rumen gases, and with maize gluten meal, which is a more degradable, more broken down in the rumen protein source, we were seeing more hydrogen sulfide, where soybean meal is more resistant to breakdown, and we were hardly seeing any increase in hydrogen sulfide concentrations. Just coming to the end, I'm just wanting to sort of wander through some of the wacky things that we've done with it, just to give you an idea of the capabilities of it. And this really links with some of Mark's last slides. These are pictures of rumen boluses. So this is what grass looks like after a cow has chewed grass and masticated it and mixed it up with saliva and swallowed it. And you know, if you take grass and rub it between your hands, there is a reaction that takes place and produces what's called green odor. The polyunsaturated fatty acids in the grass are broken down by lipoxygenase and fatty acid hydroperoxide lyase to produce particularly aldehydes and alcohols, and this really does link very much with Mark's last slides, with the tomatoes and the mangoes. So they're being produced by the grass as a plant defense compound, but we got quite interested in how that affected the colonization of grass by microbes in the rumen. We developed a model. Again, this is when I was working in New Zealand, where we use freezing and thawing of grass to simulate the damage that occurs, the cellular breakdown that occurs when you during mastication, during ingestion of grass, and then use the Sift MS to monitor the release of these so-called green odor compounds. So a lot of them from fatty acid breakdown, hexanal, hexenol, hexanal acetate, but also a lot of methanol, which is coming from particularly some of the pectin within some feeds. So yeah, that was just thrown in as another example, and that was a situation where you could use the kind of auto sampling capabilities linked to Sift MS to control that kind of experiment. So you froze the grass, and then as you thawed it, you were able to monitor the release of volatiles during a controlled thaw process. So I think I'm being the chairman's friend and have finished there, so I can hand back.
Thank you very much, Richard. That's excellent. Yeah, we are going to take questions, but we're going to take questions in probably 3 or 4 minutes. I've just got a last slide to show all of our attendees today, just to round everything off, really. So if you can see my screen there, I just wanted to let all of the attendees know that Agri-EPI actually do own a selected ion flow tube mass spec, so a Sift MS machine. So as we've heard from our speakers today, this is one of the leading analytical techniques for comprehensive gas analysis to ultra trace levels to analyze volatile organic compounds. So it's a precise and quantitative analysis with unparalleled selectivity and reduced pre-concentration or sample prep. So I've just put some examples there of the sort of projects that this piece of equipment could be used in. So as we've heard, the detection of oxidized fish oils in an aquaculture project, detecting volatile compounds from cows in livestock, and to detect flavor components in a range of foods, or headspace analysis in food packaging, just some examples there. So Agriepis Sift MS is based, or going to be based, in our Northern Food Lab at Roslin, so on the University of Edinburgh campus. And it will be available from January 2021, and it will be fully serviced with a trained on-site technician. So this piece of equipment is available to rent or to partner with Agri-EPI to use as a subcontractor or a partner in either a CR&D or a commercial project, if you so wish to use this piece of equipment for your agri-food project. And it is complemented by Agri-EPI's portfolio of technical and on-farm assets and services. So, and it is the only UK-based Sift MS specifically available for agri-food applications. So if anybody is particularly interested, then please do get in touch. So we have come on to question time. I'm just going to open up our question box and see what we've received. So fantastic. We've got a question here from Rachel Wakefield. So thanking everybody for your presentations. Is there a reduction goal for volatile organic compounds from livestock? And also, is there a critical subset of gases to monitor that would be an index of others that may be present? So I'll split that into 2. Richard, if we could go with you first to answer the first question, that's, is there a reduction goal from volatile organic compounds from livestock?
I'm probably not the ideal person to ask on that. I mean, so there are gases that we know about, like methane and nitrous oxide, that have that contribute to the greenhouse gas national inventory, and so that, you know, those are important and monitored nationally. I think in terms of emissions from livestock, it's probably the best example would be in terms of the manures and manure handling, and you know, that's an area where there would be interest from the public and authorities in relation to nuisance. So that, you know, so many of this, you know, the sulfide compounds in particular, I think would be relevant there in terms of, you know, the consequences of spreading manures inappropriately. I mean, I think it all kind of interacts a bit with the greenhouse gas story as well, because I think and again, so we've talked about methane and nitrous oxide. I think ammonia emissions is another thing that's rapidly increasing up the agenda, which I don't think is, well, I think that the Sift MS, I understand, could separate out some of the amines that are associated with that. You know, I think probably in terms of understanding volatile emissions from manure, to know about the amines and the sulfides, I think would be valuable. But I'm not a manure scientist.
Yeah. No, absolutely, Richard. I mean, you're right in terms of ammonia coming to the forefront and looking at policy changes, and the clean air strategy, especially sort of coming into force in the UK, is becoming more and more important to monitor volatile organic compounds in agricultural settings, and the Sift MS is an ideal tool to be doing that, both in research and commercial settings. Second part of the question from Rachel, that I'm just going to sort of hand over to Mark. It says, is there a critical subset of gases to monitor that would be an index of others that may be present?
That's a very interesting question. I think, again, going back to what Richard said in terms of, you know, odors from these areas, when you look at those compounds that particularly are problematic, I think if you monitor things like ammonia and the small volatile fatty acids, interesting enough, those compounds I think that would be a very good marker for a more broad you know, release. Things like the small sulfur compounds, the small amine compounds, so ammonia, hydrogen sulfide, etc., are always the ones that are most difficult to analyze by traditional techniques. And in fact, there was a presentation put together by a colleague at Sift about a wastewater treatment plant, where they correlated very strongly the release of things like hydrogen sulfide, dimethyl sulfide, and trimethylamine more generally with the odors being given off by these wastewater treatment plants. You could actually make a very good correlation of it, and in a way they became quite good marker compounds for everything else that was happening in that plant, because they're the ones that have the strongest odor, you know, threshold values for humans. And so that's really what you're looking at in the end, is for these nuisance odors, it's what compounds have the most activity from an odor threshold perspective, and they're always the ones that are really difficult to analyze. As an analyst going back 20 years, these are difficult compounds, but Sift MS actually makes them quite trivial. I mean, going back to ammonia, it's easy to do ammonia with Sift MS, surprisingly easy to do ammonia with Sift MS, given the compound it is. And so I would argue, yeah, these small odorous volatile compounds can act as really good surrogates for a more general process that's taking place.
Excellent, thank you very much, Mark. So we've got another couple of questions here as well. One for you, Richard. How do you anticipate Sift MS being used for animal science applications in the instance that it can't be taken into animal sheds?
I think I mean, I think I kind of hinted at it in the presentation. I mean, I think, you know, there are, I think Mark has given examples where the time course of things becomes important, but I think another area of application is where you're kind of scoping out low-cost sensor applications. I think in you know, particularly in dairying, in robotic milking, I think we're wanting to have low-cost sniffer sensors. You know, we're not. I mean, you know, a Sift MS costs more than a robot milker, so we're not going to have a Sift MS on every robot milker. But I think, you know, I think there probably are sensor technologies that we want to include in that. And I think, you know, some of the ground truthing and understanding the interactions of gases and things, Sift MS will help in identifying that.
Excellent.
I mean, honestly, I think I mean, one of the scary things of Sift MS is the amount of work that you can get through in a very short short amount of time. So I think probably we do need to explore more of these time course kind of applications. You know, if you.
One point I was going to make regarding sort of the remote monitoring or remote sampling is that one of the things that we've done over the last few years is really optimize the automation that sits on top of SiftMS to the point now actually that we can do headspace samples once every 2 minutes on this system. We can actually do thermal resorption tubes. And one area in human health, which is really growing, is the idea of breath analysis, particularly breath analysis onto thermal resorption tubes as a way of shipping samples to a central site to analyze it. And so you need to do sort of remote sampling into a bag. TD tubes are brilliant for that. You know, you can do multiple samples on thermal resorption tubes, and we can run through 500, 600 samples a day with a SiftMS for targeted analysis. So you can imagine, comes back to the idea of data set size and data density. You can get a lot of information if you sample, even remote sampling, and bring it back to a centralized source, centralized hub. It works really effectively.
That's great to know, Mark. I mean, the Agriepis Sift MS is going to be based up in Edinburgh, but that by no means means it isn't useful for projects that may be going on in the south of England or, you know, far further north in Scotland. So no, that's brilliant.
I've received bags of cow breath into the lab here in Cambridge from Edinburgh, so I know it works. You know.
Yeah. Exactly. We do have one more question from the audience. I think we've got time. We've got 5 minutes left. This is from Andy Evans, and judging by the question, I think it's going to be aimed at you, Mark. Can volatiles collected on SPME fibres be used in SiftMS?
That's a very interesting question, that, because it is something that we have looked at, and the reason we've looked at it is as I say, it gets back to this ability to do automated samples. So between us at Anatune and Sift, we've developed some injector heads that can sit on top of SiftMS. They can take things like thermal resorption tubes, can take injections from headspace vials, and it should also be able to work with SPME fibres as well. And we've looked into this, and the thing is that we don't know exactly how effective this will be, simply because it's a capacity issue on the SPME fibre. Is that, you know, it's a difficult one to go. But in principle, yes, it can. There's no reason to say it won't, but I think there are probably more effective ways of doing it. In fact, one of the reasons people do SPME as a technique, particularly for GC, is to do with a sensitivity issue. They're not achieving the sensitivity from a single headspace injection or whatever, so you concentrate on the SPME fibre, and then that gives you that sensitivity gain in the instrument. Interesting enough, SiftMS has that low end sensitivity anyway, where actually SPME probably doesn't offer as much of a benefit as you might imagine, because whatever you're concentrating up, you're just, sounds bad to say, wasting a bit of time, because we have the sensitivity at the bottom end anyway. We can do sub parts per billion concentrations with SiftMS quite straightforwardly. So in principle, yes, we've looked at it, we've looked into it. We would go with thermal resorption tubes first as the first thing to do before we do SPME, but yes, it's an interesting question. We think about this quite a lot here, doing this sort of stuff. We've got the technology to do it. We've just never really done it properly, that's all.
Well, I think we might have the perfect partner, some in Andy Evans
yeah, exactly. We're always looking for that sort of partnerships to develop this stuff. If we've got somebody who's interested in it, we'll absolutely take it on, because, you know, it's a good driver to get these things done. It's somebody who's actually interested in what you're doing.
Yeah, fantastic. We've run out of time and we've run out of questions. It's been fantastic to have you both on the call today, and thank you to all our attendees for listening in. We'll be reaching out to all of you individually. We really hope you've enjoyed the presentation. Please do follow us on Twitter and LinkedIn, and then you can see all the new news coming through about our webinars and events. So that's it for now. So just a final thank you, especially to Mark and Richard and the team at Agri-EPI who are helping me in the background here as well. So thank you very much, and we'll all sign off from there.
Thank you.
Bye.
Cheers. Bye all.
Bye.